everyone. Welcome to the US MAP2C Foundation Scientist Spotlight Series. I'm Rachel Pignotti, Marketing Strategist at the Foundation. We're excited to introduce Dr. Jayne Aiken, Assistant Professor in the Department of Biomedical Sciences at Colorado State University. Dr. Aiken's discussion topic today is harnessing the power of Simons Searchlight. Few things to go over. The webinar is being recorded today. Please use the Q&A for all questions. We'll address those at the end of the presentation. We hope to have time to address all of your questions, but if not, they'll be addressed offline. Following the webinar, please direct all questions and comments to the USMF2C Foundation, organizers of this Scientist Spotlight Series, at info@usmf2cfoundation.org. Thank you. Dr. Jayne Aiken, would you like to take it away for us?
Absolutely. I'm just going to start sharing. We'll see if we can get my
Perfect.
slides
All
here.
right.
Well, thank you so much for hosting me and having me come present for you in your webinar series. As Rachel told you, I'm a new assistant professor at Colorado State University, and I have experience studying neurodevelopmental disease genes from the perspective of the basic science, understanding how these mutations to specific genes alter neurodevelopment, and also marrying that data with the phenotypic clinical data that's assessed for different patients and bringing those two data sets to get a better understanding of disease progression. So today there are a number of areas that I'd like to discuss today. I'm going to talk about the power of iPSCs. We'll get all into what that means if you're unfamiliar with that term. I also am going to share data from the example that of data that I've gathered looking at KIF1A, which is another gene like MEF2C that leads to neurodevelopmental disease and causes changes during cellular neurodevelopment. And then finally, I'm going to end with the potential for MEF2C and the directions that I'm going to take the Simons Searchlight resources, such as patient samples, as well as the phenotypic survey data collected to help understand MEF2C related disorder and help us as a community understand ways forward with that disease. So first up, we're going to be talking about iPSCs. And I like to start my presentations by getting everyone on the same page. So I like to start basic with a cell. Many people may have known from, just around or middle school biology class, the cell is the basic building block of the body. And I like to use the analogy of Legos. So most people are familiar with Legos. You have your little blocks and you can put them together. But in order to build really complex, fun Lego structures, you're going to need more varying types of Lego bricks, different colors, different sizes. And with these, you can put them together to make your Lego masterpiece, such as this beautiful Bob the Builder. And that holds true for the cells in the human body. You have different types of cells that are doing different things. You've got skin cells, muscle cells, brain cells, such as neurons, which we'll be talking about later. And all of them have to properly be formed during development and come together to form an organism or a living being. And so in my Lego analogy, you can see how these different layers of complexity come together to build the human. This is a sciencey depiction of what I was just talking about, where you start with a stem cell. It can become many different types of cells, and all of those are necessary to build the human body. And an exciting, relatively new and very revolutionary new technology that's been introduced in the last few decades is the idea of the induced pluripotent stem cell. So that's what the acronym iPSC means. And so what an iPSC is researchers are able to take differentiated cells, so for example, cells from the skin, and de-differentiate them into something that looks like a stem cell. So you can take a sample from a patient, make it back into a stem cell, and from that cell, you can make all of the different types of cells in the body. And this is a really powerful tool to study human disease because using that specific patient sample with all of their genetic information, we can then harness the iPSC cell into different types of cells. And for today, I'm going to be focusing on neurons because neurons are the major cell in your brain and throughout your body that are important for sending different communication signals that allow us to think, move, and everything in between. So I am going to be focusing on the neural cells. And that brings me into the next phase of my talk here, where I'll be talking about research that I've done studying variants in KIF1A. So, as I mentioned, neurons are extremely important in your body. Your brain consists of billions of neurons all connected to each other. The neurons in your brain and along your spinal cord make up your central nervous system. But then there are neurons all throughout your body too that connect to those neurons in the central neuron and out, out to the tips of your fingers and to the tips of your toes. And so you can imagine that all these neurons are connected to each other, talking to each other to make us able to do all of the amazing things that we can do. And within a neuron, you can see I've got a little schematic of what a cartoon of what a neuron look like. Unlike that little round cell that I presented at the beginning, neurons are really unique because they have the ability to extend out these long processes, these long arms, which we call dendrites and axons, that allow them to reach their targets. And that's really important for them to communicate properly with one another. And if we look inside those little neuron arms, there's important things happening that allow things to zoom around the cell. And one of the things that I'm particularly interested in as a researcher is a structural element of the cell called the microtubule. So the microtubule is part of the cytoskeleton, which means the cell skeleton, and it provides structure for the neuron, and it also acts as tracks for little motor proteins to walk along and distribute things throughout the cell. And that's really important because a lot of the things that a cell needs are made in the cell body, the little round part of the cell, and they need to get distributed out all the way through this neuron for it to be healthy and perform all of its functions. And that KIF1A gene that I mentioned earlier happens to be one of those motor proteins that walks along the microtubule to distribute cargoes throughout the neuron. And so I was particularly interested in how mutations to KIF1A can disrupt its ability to perform its job of moving things along the microtubule. I love showing this video. This is from the Inner Life of the Cell video that was generated at Harvard years ago, but it's still one of my favorites. And this helps you understand the really dynamic nature of these proteins inside of a cell. So here you can see a microtubule building itself, disassembling the really dynamic polymers. And then my favorite, the little kinesin motor walking along the microtubule, carrying its cargo throughout the cell. I mean, I just think it's adorable. And so that's what a kinesin is doing inside of a cell. And I wanted to understand how, when we have these variants that have been identified in KIF1A, that in these different patients, how that gives rise to developmental and degenerative problems. So here's some example of the KIF1A community that I work with. Like MEF2C, KIF1A encompasses a wide variety of clinical phenotypes, from changes to how the brain is forming, all the way to how the motor neurons are connecting and how the kiddos can move and think and communicate. And I came into this project trying to understand how different mutations to KIF1A could alter the neuron function during development. And so this is one way to look at one way to classify different variants that occur. And many of you may be familiar with looking at a diagram like this from your experience with MEF2C. And you can see this is like a little cartoon schematic of the protein, and all these little lines and letters that come along here represent specific patient variants that have been identified in the community. In KIF1A, there have been many, like MEF2C, that occur throughout the protein. This is really useful, but I also wanted to come in and characterize or classify the mutations in a different way, which was based on how they were predicted to disrupt the protein function. And so this is a schematic that I made for the KIF1A community, and you can see all of these specific variants are classified into groups based on what they're predicted to do to the motor function. So as I introduced earlier, KIF1A normally walks along the microtubule to distribute cargoes throughout the cell, but there are all different types of ways that that can be disrupted. It could be not able to bind its cargo. It could be not able to bind the microtubule. It could bind the microtubule but walk too slow. It could bind the microtubule but walk too fast. There are all these different categories of ways to disrupt the mutation. And I wanted to create a resource to help understand all these different categories by picking one representative mutation from each class and studying that in a cellular system. And so to do this, I worked with KIF1A.org as well as Jackson Labs to pick out a panel of mutations that represent different disruptions to the protein function. And that's what I'm going to be talking about today, where we look at what happens when the motor is not able to walk fast or far enough, when it goes too fast and too far. That's the blue. And then another really interesting category that leads to the most severe clinical phenotype, which is called a rigor. So it binds to the microtubule and it can't let go and it can't walk, which you can imagine is a really bad gain of function, as well as understanding what happens to a cell when you don't have any KIF1A at all. And so that's this truncated protein here represents we don't have any functional protein left. And so using these stem cells, which were made by Jackson Lab, I'm able to differentiate them into neurons. Here's an example of how I do that. You can see that by 3 weeks, I can induce the differentiation of neurons that look like the cortical neurons in your brain and perform intricate studies on how they're functioning. One of the things we can assess is how well neurons extend those arms out that are important for connecting to one another. And we were somewhat surprised to find actually that all three of these mutations that lead to a diminished ability of KIF1A to function decreased the length of these neurites as they're extending at early stages, while that really fast KIF1A mutant did not alter this. Another thing I wanted to look at specifically was how a particular neuronal cargo is distributed throughout the cell. So a while ago, KIF1A was discovered. It was 1995, and actually the very first cargo that was identified to be important for KIF1A was synaptic cargos or synaptic vesicle precursors. And so I wanted to look within a live cell and watch how these cargos are being distributed. And so you can see here in this movie, little things zooming around inside a human neuron, which is pretty amazing. I still get excited about it when I'm sitting there imaging these. And we can create a map of where these things are, where they're going, where they've been, which is called a kymograph. And you can see that these lines that are streaming across represent cargoes that are moving, ones that are stationary up and down represent accumulations. And so we can look at all these different KIF1A conditions to see what happens to the movement. And you can see in wild type, you've got these nice green movements. With the hyperactive, where it goes too fast, too far, you can actually see that happening. These cargoes are streaming by faster than normal, and there's a lot of them. Whereas a more extreme example on the other side is that rigor mutant that binds the microtubule and doesn't let go. You see very few cargoes moving in this case. And so we can take that live imaging data and we can quantify it. So we can count things and plot them, which is, as scientists, our favorite thing to do. And you can see that in the case of the hyperactive, they're going faster, whereas with the Riger mutant, they're going slower. Same for that hypoactive, they're going slower. We can also look at how many things are going, and you can see that that same trend holds through. So with these different variants, it's affecting the way that these cargoes are moving around in the cell based on those characteristics of that KIF1A mutant protein. So with that data, I was really interested in understanding, with this change in motility of different cargoes in a neuron, how does that change where things end up? So you can imagine you've got that little round cell body of the neuron, and then you've got its long extensions. In particular, I'm interested in the axon, and I wanted to see where things are. Are they stuck in the cell body? Are they out in the axons? And so to do that, I can specifically look for these proteins in different places using antibody staining. So there's a little antibody that will recognize the protein and we can visualize it. So you can see here, this is the soma of a normal wild type cell that doesn't have any mutations to KIP1A. You normally get some of these presynaptic proteins in that soma where they're being produced. But when we alter KIP1A function and decrease its functionality, its ability to move, you get more of those proteins stuck inside the cell, which is quite dramatic and obviously not very good for the function of a neuron. Whereas in the opposite case, where it's going too fast, you actually see fewer of these cargos in the cell body. If we look down the axon arm, you can see that the opposite holds true. So in these like loss of function situations, you're getting less out in the axon, where the hyperactive, there's more getting shunted out there. And then finally, I like using this assay where I can force a neuron to make a presynaptic compartment, and I can see how much of the presynaptic stuff is getting there in these different conditions. And you can see that, again, the same trend holds through. You're getting less of your presynaptic cargoes at these presynaptic formations in these three loss of function conditions, where you're getting more in the gain of function. So this was all really interesting, but I wanted to be able to tie the cellular data to more functional data. And a neuron's function is to send synaptic signals to its neighbors. And so we're able to assess its ability to synaptically fire using something called a multi-electrode array. So we grow the neurons on this grid that detects electrical activity. And in this movie, you can see there are these little spikes when a neuron fires, and you can see it in real time. And this is really exciting. I'm a cell biologist at heart. I'm not really an electrophysiologist. So the fact that I can grow these and get this kind of data is really exciting to me. And we can quantify this, and we see that, again, we're seeing this different grouping of variants in how they're reacting, where with these three loss of function variants, you're getting a decrease in its ability to spontaneously fire. Whereas really for that hyperactive mutant, in the assays that I've done, it's not really altering its functional ability, which holds true with the phenotypic data collected from patients, where the hyperactive have less severe clinical manifestations, and the Riger has a more severe, and that's holding true with the cellular data as well. So here's just a little graphic to reiterate what I've told you just so far. We've been able to characterize these different classes of mutations to gain insight into what they're doing to cellular neurodevelopment. And I'm really excited to continue to explore how these disrupt overall cell function, and also use these as a model to test specific therapeutic strategies, which need to be different dependent on the different classes of mutations and how it's altering the function. So finally, I just want to end our time talking about the potential for MEF2C and how I'm going to be using the data set that is being collected by Simons Searchlight to perform similar assays for the MEF2C community. So like KIP1A.org, I feel very fortunate to be working with a wonderful foundation such as the US MEF2C Foundation, and really get in while I'm beginning my own research in my lab, make this an important project and focus. So like KIP1A, we see time and again that different mutations that alter neurodevelopment give rise to a broad phenotypic spectrum of different clinical manifestations. I've listed some here. And it's really important for us as researchers to understand how different variants present in different patients, because linking that molecular change to the behavioral and developmental consequences in a patient is really important for understanding its role in its disease progression. And MEF2C is a particularly complex case because, unlike KIP1A, which encoded a specific protein that we knew quite a bit about what its role in the cell was, MEF2C is a transcription factor, which means it actually regulates what a cell decides, what proteins the cell decides to express. And that's really important because if we bring it back to that Lego idea at the beginning, what proteins a cell expresses controls what type of cell it's going to become and how that cell functions. And so I was particularly interested in MEF2C because when I looked at the target genes that it regulates, so MEF2C will come in and tell a cell, turn this gene on, turn this gene on, turn this gene on, et cetera. When we look at, there's like almost 2,000 genes that MEF2C regulates. If you look at all these ones in green, a lot of them are regulating cellular transport, which is what I am an expert in, and it's incredibly important for neurodevelopment. And so, what I'm really interested in doing with the MEF2C resources is understanding how particular variants change the protein landscape of a cell, and then how that in turn changes the organization and transport of cargoes throughout a neuron for different MEF2C variants. And so that's why I'm currently working with Simons Searchlight to turn patient samples that have been collected into iPSCs that I will bring into my lab and perform cellular assays on, as I did for KIF1A, as well as link those important molecular and cellular insights back to a really robust uh, longitudinal clinical data set that is being collected by Simons Searchlight that will help us understand how specific variants are manifested in a patient over time. And that's going to be really important for understanding the basic molecular mechanisms at play within a cell, as also for exploring specific therapeutic strategies that will be relevant across these different variants. And so, with that, I just like to thank you guys for your attention, as well as my institutions and funding. All of the work that I showed you today was performed when I was at the University of Pennsylvania, but I'm currently moving my lab to Colorado State University, where I'll be continuing looking into MEF2C.
Dr. Aiken, thank you so much for your time today. I love your logo here in the center of your
Thank
slides.
you. The Aiken Lab.
It was really interesting to hear about your prior work and how it's going to tie to what you're looking at with MEF2C. So that was a really nice illustration for our viewers, I think. We don't have any questions in the Q&A right now.
All right.
So I think you provided quite a bit of information for us. But we did explain to our viewers that they can reach out to us at the info at usmef2cfoundation.org email address. They can also contact us through our website as well. And any questions that we receive after the presentation, we can pass along to you. But for now, I think that concludes the presentation. And thank you everyone for your time. And special thanks to Dr. Aiken and her team. Thank you so much.
Absolutely. Thank you for having me.
Thank you so much.